Right, so welcome back everyone to the sort of second part in this guest lecture series on single-cell RNA-seq analysis. And so today I will pretty much start off or pick it up where we left yesterday. So I'll first be talking about, on Tuesday, where we're talking about how to do some dimensionality reduction, feature selection, and visualization of your single-cell RNA-seq data. So first of all, something you may have heard of, because it's something which is not just for single-cell RNA-seq, but pretty much any other types of data where you have really high dimensions. This thing called cursive dimensionality. So very loosely speaking, what that means is basically that when you get into very high dimensional space, sort of distances, be it Euclidean or really any other type of distance that you want to consider between data points, sort of become less informative. So what that means in practice is that sort of pretty much wherever you are, every other data points look to be sort of more or less at the same distance. So distances are no longer as useful as they would be in lower dimensional space for sort of discrimination and other tasks. So one example is sort of shown here in this plot here. So here we took a small data set on early mouse embryonic development, and we consider these sort of two different scenarios where you either use sort of all 20,000 genes that were profiled in the samples, or you sort of use a selection of the most variable genes, only 500 of them. And as you can sort of see here visually, you have both sort of the clusters are sort of more distinct here on the left compared to the right. And you can also see this sort of quantitatively here, that the fraction of variance explained by the different principal components is higher here. So, so this reduction of the number of features has sort of allowed us to sort of better pick out the sort of the biologically meaningful structure in this data set. So this is sort of a very useful strategy for dealing with this problem of the cursive dimensionality. So this is a sort of example of what's called feature selection. So by feature, we mean gene here, really. So the idea is to sort of not consider all of the genes in your sample, but really only a subset. So one of the things that you always should do is sort of just look at for the genes that are just really lowly represented. Sort of typically when you would use this sort of a threshold of a few percent. So if a gene is present in no more than a few percent of the cells, then you just take it out altogether. So that can typically reduce the number of genes by sort of a few thousand. It really depends on how deeply sequenced it is. But then you want to go sort of further than that. And one strategy, which is the most popular one in the field, is to look at sort of highly variable genes. So it is, of course, the case that the variance is proportional to the mean. So we can't just look at the variance per se, but instead we look at the normalized variance of a coefficient of variance, variation, which is sort of shown here. And so here's a sort of a plot from where this method was published, which, as you may see, is actually very long ago by single-cell RNA-seq standards. So what you can see here, each dot represents a gene, and then you look at the mean expression versus the normalized variance. And we're interested in the genes that have sort of so the red line here represents sort of the mean, the average trend, and we're interested in the genes that are higher variance than you'd expect by this trend. These are the pink ones that show up here. And so what's worth pointing out here is that the blue dots correspond to these ERCC spike-ins. So these are sort of artificial spike-ins that have been added to your sample. So spike-ins is really something that was sort of very popular in sort of Jurassic era of the single-cell RNA-seq field, which would sort of correspond to like 2014 or 2015 or so. So you may have to ask a sort of senior postdoc in your lab what they are and why they're useful, but they've really sort of fallen out of favor these days for various reasons. Hence, I'm not really talking about them. But these are supposed to be sort of negative controls, and as you can see here, none of them are highly variable, which is a nice thing, a nice confirmation. Another strategy is to, instead of focusing on the genes that have high variance, is to focus on the ones that have high levels of dropout, so more zeros than you would expect. So the trend that you'll see in sort of pretty much every data set that I'm aware of is that you have a very strong correlation between the dropout rate, so the fraction of zeros, and the average expression. So what that means is that if the gene is highly expressed, you have few dropouts, and vice versa. And you can sort of plot this, and you can sort of fit this type of sigmoidal curve here. And then what we're interested in are the genes that are sort of illustrated here in purple. So these are the dots that are sort of have a higher number of dropouts than you would expect by chance. So those we can pick out and use as features instead of the highly variable ones. These will to some extent overlap, but they're not the same as the highly variable genes. So another strategy that we can use to deal with the curse of dimensionality is dimensionality reduction, which is a very big field in itself. And there's been lots of work done on that in statistics and mathematics, and lots of methods that have been applied to single-cell RNA-seq. And the reason why dimensionality reduction works here is quite straightforward, right? Because so you have 20,000 genes, but then you can imagine, for example, that you have one transcription factor which controls sort of say 50 other genes, right? Then in reality, those 50 genes don't have 50 separate dimensions, but it can describe by one dimension, which represented by this transcription factor. So this sort of, more formally speaking, what we have is a so-called manifold hypothesis, which is that there is this sort of lower dimensional structure embedded in this higher dimensional space where all of these points, the data points, the cells actually live. And so what we're trying to do is identify this low dimensional manifold, and then we can use that to represent the data instead. So this is of course hard to illustrate in because you can't really draw more than 3 dimensions, but this is sort of very famous example for illustrating this. So this is called the Swiss roll. So you can see here that we have this sort of structure which sits in 3-dimensional space, but in reality it's actually a 2-dimensional surface. So ideally your method should be you only require two coordinates to describe this. So ideally your method should be able to identify that we have a 2-dimensional surface sitting in 3-dimensional space. So there are multiple methods for trying to deal with this. The most popular one by far, and is really an old workhorse from statistics, is called principal component analysis. Really, the main advantage of principal component analysis is that it's linear. There are sort of very efficient methods for calculating, even for large data sets, and it's easy to interpret what it means because the different directions that it picks out are the ones that explains the most variance. So we can easily interpret distances and also angles and positions in the PC space. So that's a big advantage of it. Fortunately, it doesn't really work that well for when you're trying to do visualization. I think the main reason is, again, sort of intuitively speaking, it can often get sort of quite dominated by sort of a few outliers. And what that will result in is that most of your data points will sort of go into one sort of small bunch. And they will sort of sit right on top of each other. So it's very hard to sort of make out any interesting structures that's happening here. So PCA will work well for really small data sets, so up to a few hundred cells. But as soon as it gets larger, you'll probably need to resort to some sort of nonlinear method to better sort of position your points from the high dimensional space into the 2-dimensional plane. So there are lots of methods around for this. What was initially the most popular one is called T-distributed stochastic neighborhood embedding, or TSNE for short. And so it's a sort of it was developed sort of in the more general machine learning field, and it's been around for quite a while, and it's very popular, and it does its job really good in terms of sort of putting the points down into the plane. It has some issues. So one of them is the S, which is stochastic, which means that when you run it different times, you'll get different outcomes. And then a bigger problem, however, is that it has a free parameter, which is called perplexity. And the outcomes, as you can see here in this figure, is that the outcome will sort of be highly determined by how you choose the value of your perplexity parameter. So here's sort of the real structure of the data points, and here's sort of how they look after going through the t-SNE algorithms for different perplexity values. And one of the issues is that there's no procedure for optimizing or how to choose your perplexity value when you run this. So you can almost make the data really look sort of whichever way you want it to choose. So that's quite problematic. And it's what's very important to stress here about t-SNE is that it is really only designed for visualization. And you should really not try to do sort of anything with the output beyond sort of looking at it, and not sort of try to interpret things the way you would interpret them using a PCA. But I think, unfortunately, that is not something which is always heeded by people in the field, and you can often sort of I feel people can sort of start discussing t-SNE plots as if they were PCA plots, and which is not the right thing to do. So t-SNE has a little bit fallen out of favor in the field. There's this newer method called UMAP, which sort of pretty much does the same thing as t-SNE. It also has like a sort of it has like this approach for doing this is based on sort of graph theoretical properties or doing a graph analysis. They argue that it's better at preserving the global structure, because one of the things that these two methods do is that they tend to focus on sort of local structures and make sure that they are sort of guaranteed to be well preserved, but there's fewer guarantees about the global behavior. And UMAP claims to be doing better. I'm not 100% sure that I've seen any really strong evidence that that is the case, but again, it's what people are using mostly these days. There are other methods. Another strategy is called sort of force-directed layout or graph layout. It's again a method which goes back quite a long time, sort of several decades, been around for a long time. The idea is very simple. You sort of think of your data points as being in this graph, where each point, the pairs of points are connected by springs. And then you essentially have a mass spring network, and you sort of let that equilibrate, and that gives you the layout of your data points. So one advantage of this is that it gives you much more reproducible results. So as you can see here is that you have sort of 3 different samples from the same type of tissue, and this sort of spring method with a forced graph layout sort of gives you sort of more or less the same topology every time, whereas when you run it with t-SNE, it looks quite different every time. So that's obviously a very big advantage here. I think the drawback of this method is that it can be computationally very costly to run it. So if you have a large number of cells, this can be problematic. So I think this is the end of the first part. Stop recording.